Hello everyone, welcome to today's live broadcast, Cost-Benefit Analysis of Expanding Bone Marrow-Derived Mesenchymal Stem Cells Using Automated Cell Expansion Methods versus Manual Methods. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by Terumo BCT. Terumo BCT is a global leader in blood component, therapeutic apheresis, and cellular technologies. It is the only company with a unique combination of apheresis collections, manual and automated whole blood processing, and pathogen reduction technologies. Terumo BCT believes in the potential of blood to do even more for patients than it does today. This belief inspires the company's innovation and strengthens its collaboration with customers. To learn more about Terumo BCT, please visit www.terumobct.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answer is welcome too, and you can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Athena Russell, MT, AAB. Athena Russell is the lead medical laboratory scientist of the Human Cellular Therapy Laboratory and assistant professor of laboratory medicine and pathology at the Mayo Clinic in Jacksonville, Florida. She has held multiple positions at the Mayo Clinic, concentrating her work on mesenchymal and hematopoietic stem cell research and other cellular and cell-derived therapies. Athena also holds elected positions with the International Society for Cellular Therapy and the American Association of Bioanalysts. I will now turn the presentation over to Athena Russell.
I'm Athena Russell. I'm from the Human Cellular Therapy Laboratory at Mayo Clinic down in Jacksonville, Florida. I appreciate you all being here for this webinar today, and it's a pleasure to be able to talk to you about an analysis that we did in our laboratory over the course of about a year or so as we were considering transitioning away from using manual methods to expand bone marrow mesenchymal stem cells towards a more automated bioreactor type system. And we really just wanted to know whether or not it was going to be feasible for us to do that given the resources that we had. To give you a little bit of background about our laboratory so you have some perspective, our lab was established about 25 years ago, but specifically to support the bone marrow transplant program and to perform traditional cellular therapy and hematopoietic stem cell processing activities. It wasn't until about 4 or 5 years ago that we started to develop protocols for investigational cellular therapies with funding from the Center for Regenerative Medicine at Mayo Clinic. And so we've been developing all of our GMP production protocols for MSCs and for some other newer novel therapies that we're getting into as well now. But we only have about 1,200 square feet of space to share with the bone marrow transplant team and our regenerative medicine team. And we had some recent renovations where we could take some of the laboratory and turn it into clean room space for GMP manufacturing. However, we're also not only limited in space, but we're also limited in staff. We only have about 3 technologists working on all of this work, 2 of them on the bench and 1 of them that handles all of the quality. And this is just a bird's eye view of what this 1,200 square feet looks like. We have 1 cell processing area dedicated to bone marrow transplant product processing. All of our storage space is located here, dry storage and cold storage, so all of the liquid nitrogen storage, 4C, -80 and -20 storage is all located in within this footprint. And then down towards the bottom is all of the newly renovated space where there are 2 labs that are certified as clean rooms. However, because we are so limited, we can only use one of them for true manufacturing, and the other one we still are needing to perform all of our research and development work. And these are just some images from the lab so you can see kind of what I'm talking about. The picture on the left is of the larger clean room space that we are needing for research and development activities. So it's a little bit of a larger space, and of course we have 2 flow hoods and 2 standard size CO2 incubators. And this is the smaller of the 2 rooms, the clean room space where we actually perform our manufacturing processes. This is an older picture, but at this time we now have a small laminar flow hood in the back, and we have 2 quantum cell expansion systems over on the right-hand side. And then on the left here is our MAYO cell processing workstation, which we still have, and it's really where we began when we were developing all of our manual cell expansion protocols. So to give you an idea of what our short-term goals are with respect to manufacturing, again, we're a small facility, we're an academic manufacturing facility. And so we really only have the ability to support phase 1 and phase 2 clinical trials. But what we really want to do is to be able to support those clinical trials that are initiated by investigators and clinician investigators here at the Mayo Clinic campus in Florida, so that we can promote regenerative medicine and cell therapy on our campus. So we want to be able to support the manufacturing needs of those here who are interested in running and developing INDs for regenerative medicine and cell therapy. But again, as I've mentioned a few times now, we have some pretty obvious limitations and challenges before us. We have a very limited amount of space that we have to work with, and a very limited staff that we have to perform all of the development activities and manufacturing activities. So I want to give you an idea of what our manual manufacturing protocols were looking like. We use the cell processing workstation, as you saw in the images earlier. If you're not familiar, the cell processing workstation is a standalone isolator type of system. It is a class 100 environment on the inside. Once it's been decontaminated, it has its own incorporated hydrogen peroxide decontamination system. It also has everything else that you need to do cell culture, like a centrifuge, a microscope, and an incubator. The incubators are modular, so you can add additional incubators if you need to, but we don't have the space for that, so we just have one modular incubator that's only 1.4 cubic feet on the inside. Contrast that with a standard cell culture incubator that has about 6 cubic feet of space on the inside, so we are working with a much less space than what we're used to having to handle all of our incubation needs. We use multi-layer flasks, and with all of the options out there on the market, we were able to narrow it down to 2 different types that we could use with this modular incubator. Those were the millipore high flasks, which have about 1,000 square centimeters of growth area, and the Corning hyper flasks, which are T1720s. This is what they look like. And for all of our manufacturing methods, we use alpha mem and 5% platelet lysate. We've gotten away from SBS some time ago. And this is how we would do it. We would start by expanding the MSCs up to passage 1 to create a master cell bank of vials of MSCs. To do that, we were able to scale up to a maximum of 10 T1000 flasks. That's the maximum number of flasks that we can fit inside the modular incubator. In those 10 T1000 flasks, we could produce enough cells, about 200 million MSCs, to freeze down about 100 vials of 2 million MSCs per vial for our master cell bank. After that, when we're ready to create a batch of product, we could pull vials from the master cell bank and further expand them up to passage 3 to create batches of stem cell product that are intended to be used for therapy. To do that, we could scale up to a maximum of 10 T1720 flasks, and at the end of all this, we could approximately create about 4 doses of product, and each dose would have 100 million MSCs in the bag. From start to finish, which is from the raw bone marrow aspirate all the way to creating the very first batch of stem cell product doses, that manufacturing campaign would take anywhere from 6 to 8 weeks to complete. So there are some pretty obvious limitations to doing it this way. First of all, you can only fit a maximum of 10 of either type of multi-layer flask inside the incubator, so you're really limited in how many cells you can produce. The whole process from start to finish is extremely tedious, very time-consuming and labor-intensive. You really absolutely need at least 2 technologists to perform these tasks efficiently. I have done it with one before during the development phase, and it just is not feasible. You consume a huge number or a huge volume of culture medium doing it this way. Each of the Corning Hyper Flasks requires 565 milliliters of culture medium to fill up the flask completely, which you have to do with those flasks. And so you can imagine towards the end, when you have 10 Hyper Flasks, and every time you do a medium change, you need 6 liters, approximately 6 liters of media. So you can see how the cost of medium and platelet can really add up doing it this way. And it's a relatively long period of time from start to finish. And at the end, you really do not get that many doses of product. So you're putting in a lot and you're not really getting a lot out of it. So it didn't take long for us to realize that we needed to make some changes and do some things differently. So we started thinking about bioreactors. We looked at a few different systems. There were 3 different systems that we considered that were small footprint, single-use platforms. One of them, of course, was the Terumo BCT quantum cell expansion system. One of the platforms required the use of microcarriers, and the other 2 platforms, the cells grew out in a more traditional 2-dimensional monolayer type of manner. However, one of those platforms we had to exclude kind of early on for space considerations, because although it was indeed a small footprint type of platform, it still required a little bit more instrumentation to go along with the whole thing than we had space for. So in the end, we narrowed it down to 2 systems, the microcarrier system and the quantum, and we brought them both in-house and tested them for expanding bone marrow MSCs. So we thoroughly evaluated these 2 systems, and eventually we decided that we had to rule out the microcarrier system. A big part of this was the amount of involvement and the time that we were going to need to translate our current 2D system to a 3D system. Because really, if you've never worked with microcarriers before, they're just so different from growing cells in flasks, and there's a lot of steps that need to be worked out and developed along the way. And we did a lot of this during the evaluation phase. We did microcarrier screening assays where we looked at a whole bunch of different microcarriers and tried to figure out which ones were going to work best for our needs and for our cell type. There was also considerations regarding the way that you have to prepare the microcarrier. Some of them are pre-sterilized, some of them need to be autoclaved, many of them need to be pre-hydrated, and all of these things you have to think about. And in fact, we don't have easy access to an autoclave. We have to walk across the street to get to one. And so obviously this wasn't really an ideal situation for us. Regarding monitoring of the culture, you have to think about that, not knowing when the cells are going to be confluent on the carriers, and also you need to keep a certain cell to microcarrier ratio. So after a certain amount of time, you have to add more microcarriers, and you have to figure out how to know when to do that. So again, just more steps that need more development. And then there's the question of how to harvest the cells from the microcarriers, and once you do, how will you separate the microcarriers from the cells? You may need additional equipment like a fluidized bed centrifuge. A lot of the techniques, microcarrier separation techniques that were on the market at the time when we did this evaluation were designed for very large-scale applications and not really for processing volumes in the 5-liter range, which is where we were. And then after all this, how can you do your purity testing? How can you be sure that you have no microcarrier contamination in the final product? So again, these are all things that we could have worked out over time, but each one of these steps needed to be developed and tested, and it would have taken a very considerable amount of time for the 2 technologists that we had doing this work. So again, we eventually had to rule this platform out. So we narrowed our options down to the platform, but we still really weren't sure if this was going to be right for us. There were some obvious benefits to using the quantum, like the small footprint. with the limited space that we have, and also the relative ease of translating our current protocols to using the bioreactor. There was a reduction in the amount of time it took for us to expand the cells, and also we were able to, after doing a number of pilot runs with Terumo's help, get to where we could produce greater cell yields in the bioreactor versus using flasks. And we were also able to keep the cells at a lower passage for banking and for production of MSC products. But probably the most valuable advantage for our particular situation was the addition of automation, because it saved us so much time with performing manual tasks. And there's also the possibility of scalability with the quantum. We have a 5,000 square foot GMP facility that is being planned out right now, hopefully for this year or next year. And so hopefully when we get this new facility and we have a little bit more space, we can add more quantums. And once we're familiar with the protocol, we can just do the same protocol with more quantums, and we'll be able to get more cell yields. So in trying to determine whether or not this platform was going to be right for us, we had to ask the question whether or not this automated platform was going to measure up in every sense to the current manual protocol that we were using. So we looked at a number of different parameters, and we did the analysis, and we were able to put out this poster that we presented at the World Stem Cell Summit a couple years ago. And if anyone is interested in looking at this poster, please feel free to email me, but I'm going to go over what we found in the subsequent slides. And again, to highlight what our overarching goals were. First and foremost, we needed the ability to expand large quantities of MSCs so that we could treat patients in cell therapy and regenerative medicine clinical trials and manufacture those cells in compliance with GMP standards. And so with this analysis, we wanted to be able to compare the automated versus manual method with regard to cell yields, cost, time and effort from the technologists, and just overall ease of performing the protocols. So what we did was we started with about 25 to 30 milliliters of fresh bone marrow aspirate, and we performed the automated method and the manual method basically side by side. We isolated and expanded bone marrow MSCs using alpha-mem and 5% human platelet lysate. We created master cell banks using the automated method and the manual method, and products using both methods as well. And then we just quantified the differences. We looked at, again, cell yields. We looked at the cost of consumables and reagents needed to perform these two different methods, how long it took to grow the cells, how much effort it required from the technologists, and how many technologists we needed to do all the work. First, looking at the automated method, you're able to dilute the bone marrow and add it directly to the quantum system. You don't have to pre-isolate the mononuclear cells as you normally do. You do have to pre-coat the bioreactor with fibronectin overnight, or else the cells will not adhere. In order to monitor the culture, you look at glucose consumption, lactate generation, and you can also see control flasks at the same seeding densities just to give you a visual of how confluent the cells are to help aid in your decision as to when the cells are ready to be harvested. You can freeze down cells into vials as early as P0, but you get a lot more cells at P1, and so we typically expand up to P1 to create our master cell bank. But you will have additional vials at P0 that you can further expand to P1 to create more cells to bank, and then of course eventually up to create more products. The product bags can be generated as early as passage 2, so you pull vials from passage 1 from the master cell bank and then just expand them up one more passage to create doses. And the total area, surface area for growth inside one bioreactor is 21,000 square centimeters. For the manual method, we first isolated the mononuclear cells from the bone marrow using Ficol and then seed them directly to T1000 flasks that were not coated at actually 50 to 100,000 cells per centimeter squared. We provided fresh media every 3 to 4 days, passaged the cells when they were about 85 to 90% confluent, and we froze down master cell bank files at passage 1. We then pulled vials from the master cell bank to expand products, but we couldn't do that. We couldn't actually freeze products down until passage 3 because you don't have that many cells in your vial for passage 1, so you have to first expand them to passage 2, and then you can really scale up by passage 3. So a very important thing to understand about how we did this analysis, because we didn't do this based on using the cell processing workstation, knowing that most people don't have one. Most people are going to be doing these manufacturing processes in a clean room with standard cell culture incubators. So we wanted to sort of normalize this so that, knowing that everybody has a different kind of situation and not everybody has the same number of incubators in their laboratories, we wanted to just kind of normalize the whole analysis down to the number of cells you could produce using one standard size 6 cubic foot cell culture incubator. And so in one standard 6 cubic foot cell culture incubator, we were able to fit a maximum of 48 Corning Hyperflask T1720 into one of those incubators for a maximum capacity of about 82,000 square centimeters for cell growth. And this is what we came up with. I know this is a busy slide and I will summarize it later. But so you can see how we did it. We looked first at all of the reagents and consumables that we would need to perform each process using each method, and for all the passages that we need to perform using each method. These are the costs for the quantum and the required passages associated with using the quantum, and also the costs for expanding manually. And these are all of the parameters that we looked at. We looked at how much did it cost to run each passage that we would need to run, exactly how much time it would take to actually expand each passage, how much hands-on time it would take from the technologists actually performing tasks, and how many technologists you would need to perform the tasks. And then, of course, we looked at approximations of how many cells you should be able to expect from each of these passages, doing it one way or the other, and how many vials you can freeze down to create a bank, and how many doses you can expect to manufacture. And so finally, we really wanted to make sense of all of this data, and we wanted to kind of put it into terms that would make sense to other laboratories that were in the same situation as us, smaller laboratories that want to support a clinical trial at their institution, but that have limited resources. And so what we did was we imagined a hypothetical clinical trial that happening at our institution, where each subject is supposed to receive one dose of 100 million MSCs. And we wanted to ask whether or not it would be possible for us to manufacture the doses needed for this clinical trial, and how much would it cost us to do it. So looking at cost, what we found was that to manufacture these 100 doses using the quantum, it would cost about $100,000, or $1,000 per dose, or $10 per million MSC. However, doing it the manual way, using multi-layer flasks in a single cell culture incubator, it would cost about $200,000 to create the same number of doses. And so the costs were about double, and the majority of that cost came really from the cell culture media and cell culture supplements, because doing the manual method using multi-layer flasks that require such large volumes of media, it just adds up the cost greatly. And so the majority of that increased cost really came from the huge amounts of media that you needed to do this procedure manually versus using the automated quantum system. Looking at this in terms of time, if you have just a single quantum, it was to take about 13 passage 2 runs to create the 100 doses that you need to run this trial. If you perform all of those runs back-to-back nonstop, it would take approximately 20 weeks or 5 months to perform all of the manufacturing activities needed to generate those doses. Although it is nice to have 2 technologists to perform these tasks, if it came down to it, you really only need one to do everything, because the tasks are so automated. And that one technologist only needs to spend about 34 total hours of hands-on work to do everything required in order to generate all of these doses. In contrast, using the manual method, you would need to perform 5 runs, and each run is defined as pulling a vial from the master cell bank and expanding up a passage 2 and then a passage 3 to get your batch. If you do all of those batch runs back to back, it takes just a little longer, about 24 weeks or 6 months. The original primary culture can be done with one technologist, but all of the work subsequent to that, you really need at least 2 technologists to perform these tasks efficiently. And with those 2 technologists, each one of them would need to expend almost 200 hours of hands-on time doing all of the manual tasks required to harvest all of those 48 flasks that you could fit into one cell culture incubator. If you have additional incubator space, you can have more growth area and therefore more cell yields, but then again, you're going to expend more medium, and you're going to need even more labor to handle the increase in flasks and processing time. So you really need more people to handle that many flasks is the takeaway point if you want to not compromise the quality of your product and the viability of the cells. So in summary, we found that using the quantum automated system instead of using the manual multi-layer flask-based protocols resulted in almost a 50% reduction in the cost of reagents and consumables needed to produce 100 doses of MSCs to run this hypothetical clinical trial. And using the automated method resulted in about a 17% decrease in the amount of actual time needed to grow out this many cells. And using the automated method resulted in an 82% reduction in the number of hours needed to be expended by the technologists in order to do all of this work, and only required the effort of one technologist versus 2 or more technologists to do all of the manual tasks efficiently. And we've concluded that we are able to produce large quantities of bone marrow mesenchymal stem cells for clinical use using the automated quantum system, and that it requires significantly less cost, time, and effort on our part to do this over the manual method using certain multi-layer flasks. And that indeed is the method, the platform that we have gone with for all of our future manufacturing needs.
Excellent presentation, Athena. Thanks for bringing that information to us. We now welcome Brent Rice, PhD, from the cell processing team at Terumo BCT, who is a collaborator with Athena and now joins us to answer your questions. Before we get started with the Q&A session, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question is, did you do a comparability study between cell products grown in the flask and cells grown in the bioreactor?
Yes, indeed. Athena did perform a comparability study according to their critical quality attributes, so their indications of identity, purity, potency, and safety. And they found that there was no significant difference between the 2 populations. I should also generally say that this is something that is pursued by many and probably most of our customers. Anybody with a historical process, of course, has their defined critical quality attributes, and then they test to ensure that the cells produced by the quantum system maintain these critical quality attributes.
Great. Does your cost include disposable cost of the quantum?
Yes, the analysis that Athena provided, there are many line items, one of which is the quantum disposables, or several of which are the quantum disposables, just as she also lists the hyperflasks for the manual process.
And one attendee asks, could I get a copy of the presentation and the poster mentioned?
The slides currently aren't available for distribution. However, to my understanding, this presentation itself should be available for the next 6 months as of today on the LabRoots website.
Okay, we have time for one last question. If we didn't get to yours, we will follow up with you via email. The last question is, have cells from the quantum been used in patients, and does it require a GMP facility?
So yes, at this point, the quantum system has been used to produce many cells that have been used in a variety of treatments. Certainly early on, more of this use was in the context of compassionate use or hospital exemption. So, for example, treatment of GVHD as a third-line treatment, or again, a last case resort for that horrible disease. However, thankfully, over the years, more and more people have submitted INDs, and more of these INDs in the U.S. are coming to fruition in clinical studies. So a couple of examples would be for inflammatory bowel disease and glioma treatment. And then in the EU as well, we're seeing a similar pattern. The most significant clinical trial actually in the world is in the EU, where they've treated well over 50 patients for heart failure with adipose-derived MSCs. And with regard to the question of facility, of course, the most important thing to acknowledge there is that the delineation of what type of facility is necessary is a function purely of the local regulatory authorities. That being said, of course, having a functionally closed and automated system like the quantum system allows you some more flexibility and ability to make some very good arguments for lowering the classification of that clean room facility. So it is not uncommon. It is quite common, and even in the largest clinical trial example that I just mentioned, in that case, while a flash-based process would have to be pursued in a Class B facility in the EU, they have pursued the use of the quantum system in a Class C facility, which has obvious benefits regarding ease of use and of course the costs of the facility itself. So yes, more and more prominent clinical production and a tendency towards using lower grade clean rooms as well.
Very good. I would like to once again thank Athena Russell and Brent Rice for this presentation. I would also like to thank our sponsor, Terumo BCT, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through January 12th, 2018. You'll receive an email from us alerting you when it's available on demand and posted on LabRoots.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We'll see you next time.